This week on Waterways, increasing queen conch spawning stock through transplantation, and hormonal studies on the Gulf Pipefish. For over 15 years, the harvesting of queen conch in state and federal waters off the Florida Keys has been illegal. During those years, researchers have monitored the conch population recovery in the absence of the fishery. Unfortunately, they have found that the total number of queen conchs in Keys waters has changed very little since the ban.
In 1986, laws were enacted that prohibited the collection of all conch in Florida and adjacent federal waters. Since then, the population was very slow in recovering. We attribute this probably due to the lack of larval recruitment to the area from upstream sources. So we had to build up the spawning stock from local sources, and because the population was so low at that time, that recovery was very slow.
I don't think the closure in 1986 came any too soon for the conch population in the Florida Keys. Although we started seeing some stabilization inshore, we didn't really see the conch populations rebounding. And then on into the latter part of the 80s and into the 90s, we started seeing declines in the nearshore populations. Areas where I used to see hundreds of conch inshore, all of a sudden were depleted.
Queen conchs are found off Bermuda, the Bahamas, throughout the entire Caribbean, as well as Florida. What Bob and his team realized is that queen conch population in the Keys could not rely upon adults spawning in distant seas for an increase in local stock. Therefore, they looked for ways to increase local spawning.
We began our project in the late 80s to survey the population and see if it would recover on its own, first of all, and secondly, to examine these proactive ways of increasing the population. We have found that the population was very slow in recovering, probably because there were very few larvae being retained in the system. And that's why we took an extremely aggressive approach to rehabilitating the population through the release of hatchery animals and now through transplantation.
In initial efforts to increase spawning stock, researchers at the Florida Marine Research Institute raised hatchery animals in their lab. The conchs grown in the hatchery had extremely high mortality after release into the wild, and thus proved to be expensive. Therefore, Bob turned his focus to increasing spawning in the wild. He soon discovered that not all adults in the wild were reproducing.
What we found is that the animals that settle near shore don't develop gonads. Their gonads just never attain the ripe condition that you normally associate with spawning populations. So it is not a behavioral problem, it is actually a physiological problem. They just do not develop. Yet when they're transplanted offshore into these aggregations, their gonads develop normally and they begin to reproduce just like the wild conch that are found in these aggregations.
I guess the conch is a very prime example of how we've learned that the inshore populations are susceptible to some phenomenon. More than likely it's water quality degradation. And I think we're seeing the halo that's around the Keys that we know exists of higher nutrients, stormwater runoff problems, all the problems that we know are confronting the water quality of the Florida Keys is affecting the local conch populations.
But the Keys are not alone. Queen conchs collected in the nearshore waters of the Bahamas are also sexually underdeveloped. However, in the Bahamas, there is a migration path for the queen conchs traverse to the offshore spawning grounds. In the Keys, there is not.
One of the interesting aspects about the conch that settle near shore is that when they settle there from the larvae that are produced offshore, they grow up to look like normal conch, but they don't reproduce. And the problem is that these conch cannot get to the offshore aggregations because Hawk Channel, which runs the length of the Keys and is silty in most locations, is very poor habitat for conch. and conch cannot cross that area as adults or juveniles. So if they wind up near shore, they stay near shore and they never reproduce. That's why we transplant them from the near shore areas to the offshore areas.
Because juvenile and adult conch do not migrate between the two zones due to Hawk Channel silty bottom, Bob and his team act as a bridge between the nearshore areas and the offshore areas. By increasing the number of sexually functional adults, conch populations could recover to pre-1986 levels. This effort requires support from many local and regional organizations.
We've taken an approach with the Queen Conch Restoration Program that it should be community-based. Therefore, we use an extensive volunteer base from throughout the Florida Keys. Every day we go out, we pretty much have volunteers that go with us and work in various aspects of the project. Today, we were working in transplanting the conch, and we have quite a few volunteers with us that are helping us to collect conch from nearshore areas and transplant them into the offshore spawning zones.
The first step on the day of transplanting is to collect the queen conchs in the nearshore aggregations. These areas, which were once prime recreational fishing spots, are now abundant with conch due to over 2 decades in conservation efforts.
From previous field surveys, we have gone and identified nearshore aggregations where there are a significant amount of adult conch. We'll go to these aggregations early in the morning and collect the number of conch that we want to transplant that day. We'll tag them so that they're easily identifiable when we transplant them offshore.
While the number of conchs transplanted in a single day can reach into the hundreds, Bob and his team are always careful not to deplete the nearshore population. From previous research, Bob has determined how many conchs can be removed while still retaining a sizable population. After the conch have been collected, the tagging process begins. Each individual is given a sequential number so that the team can keep track of them in their new habitat offshore.
Each conch is tagged with not only a unique identifying numbered tag, but we also put a piece of flagging tape on, which is a different color for the different days that we go out. This signifies the transplant day so that we can quickly identify the conch underwater at the transplanted site as to the day that it was transplanted to that site.
Actually, you know what that is? That's biology.
Yeah.
2972. 2960. 22.4. 20.8.
When the conchs are flagged and tagged, they are put into containers and a wet towel is draped over them to keep them cool and moist. They are now ready for the 5-mile journey across Hawk Channel to an area of protected reef tract offshore. The transplant area is marked by a grid that allows the scientists to distribute the conch evenly throughout the zone. One by one, the nearshore conch are carefully dropped into their new marine home offshore. With the transplant complete, the work has just begun for Bob and his team. They must document and illustrate the effectiveness of the program by tracking the success of the transplanted conch.
When we go to these areas where we have transplanted the conch, we look at several things. First, we go in the water using scuba and we identify the native conch, that is the ones that are native to the offshore area, and the transplanted conch. For the transplanted conch, we look at the tag number and the color of the flagging tape so that we can determine when they were transplanted. We also observe whether or not they are engaging in copulation, which is how conch reproduce. We look at any type of egg mass production, which is where the larvae will eventually come from. And we also note where in the grid these conch are found so that we can actually look at their movements relative to their wild or native counterparts. So when these conch are transplanted offshore and they begin to reproduce, they produce these egg masses that look like croissants made of linguine. These egg masses can contain up to about 200,000, 250,000 eggs. About 1 in 8 million will survive to again reproduce. The eggs hatch after about 4 days, and the larvae then float to the surface and are entrained within whatever circulation patterns exist in the area. There are a lot of very complex patterns in the Florida Keys that may facilitate the retention, that is, conch staying within the Keys system. We choose the areas where we transplant the conch to maximize the chance that the larvae are retained in these systems.
How can Bob be sure that the larvae that spawn here will return to Keys waters? Is the increase in queen conch reproduction helping the Keys population, or are the larvae exported by ocean currents ending up in the Bahamas or Bermuda? An important aspect of the project has been to examine the distribution pattern of queen conch larvae. The team has carefully analyzed ocean currents through their diligent collection of plankton throughout the Keys waters.
We have a good deal of evidence that suggests that conch larvae that enter the Keys system originated from the Keys system. Very few conch larvae are coming in from upstream, that is, sources like Cuba or Mexico or Belize. This was done in a very limited scale, so we want to confirm that this is indeed the case, but this may explain why the population was very slow in recovering. We had to reach a critical mass of spawning conch in the Florida Keys to ensure that there was enough larvae being retained in the system and then being deposited back into the Keys.
Since the program's inception, Bob and the staff at the Florida Marine Research Institute have shifted strategies, but the goal has remained the same: to increase queen conch population in the Keys and ultimately to reopen the recreational fishery.
I've never seen such dedication from scientists like Bob Glazer and his team at the Florida Marine Research Institute in really focusing their efforts on something so important to the Florida Keys as the queen conch. I think everyone in the Keys rallies around the fact that we can't lose our symbol. We can't lose the queen conch.
The transplant program has shown early signs of success, and the project has been praised by many state and federal agencies. With a Florida spawning population of about 25,000, the queen conch is listed among Florida's protected species. While protection from harvest has stabilized the population, transplanting conch may prove to be the best method for increasing spawning stock, thereby increasing the population of queen conch in the Florida Keys.
I hear from the general public all the time that they've seen enough science. We don't need science, we need action. And I think what Bob Glazer is doing is action, but he's doing it based on science. And as a manager, I can grab that kind of science and put it to use in a management application. And it's really a beautiful convergence of science and management in such a way that we can bring the queen conch back.
Everything down here has the name conch in it. The high school in Key West, the hotels up and down the Keys, there are restaurants that are called conch, the conch train. And so conch really represents a lifestyle here in the Florida Keys. And it is our goal to ensure that the conch republic does not become the conchless republic.
Hidden in the fields of Florida Keys seagrass, there lives a creature called the Gulf Pipefish. At about 3 to 5 inches long, the pipefish can be found throughout the Gulf of Mexico, from Florida and Louisiana all the way south to Brazil in both freshwater and saltwater.
I am studying the Gulf Pipefish in the Florida Keys. I am studying the behavioral endocrinology of the Gulf Pipefish. And what this means is I'm studying the behavior of this animal and the hormones that underlie that behavior.
Sunny Scoble is a behavioral endocrinologist and visiting scientist at Mote Marine Lab in Summerlin Key. A behavioral endocrinologist is a scientist who studies how animal behavior relates to the hormones produced by its endocrine system.
Most animals have males that compete and females that get to choose. But there are a few species that are called sex role reversed species because the sex roles are opposite.
One unusual characteristic of the Gulf pipefish is that it is believed that the sex roles are reversed. The males carry the fertilized eggs, and the females compete with other females for a mate. Because the sex roles are reversed, a female must be able to produce eggs and be aggressive at the same time. The hormones involved in these two processes may or may not be the same. The study aims to find out.
I think we have a lot to learn from the Gulf pipefish and sex role reversed species. It sets up an interesting situation when you look at the physiology. We know that many males, their mating behavior and their aggressive behavior is controlled by testosterone. It's mediated through testosterone. So a male that is getting ready to breed gets very high levels of testosterone production in his blood. This gets him ready to be physiologically ready to breed, and it also gets him behaviorally ready to breed. When you reverse the situation and you've got a female, it sets up an interesting problem. You've got a female that needs to be able to produce eggs, but you've got a female that also needs to be aggressive. Is this controlled by testosterone? In some females, they've shown that high levels of testosterone can actually shut down reproduction.
Through a series of studies, Sunny will determine which hormones influence female aggression and what effect the production of these hormones has on the fish's ability to reproduce. Sunny and her assistant Jason Kim have come from the labs at the University of Oklahoma so that they can collect enough data to complete their first study. On this day, Sunny is in the field collecting some specimens. Since the Gulf pipefish is so adept at hiding in the seagrass, Sunny and Jason use a method called seining to collect the fish. The seine is a large net with very small holes that Sunny and Jason drag through the seagrass, collecting all the animals that hide among the blades. Waist deep in water, they shuffle across the muddy floor, seining through the seagrass beds. After about 20 to 30 feet, they check the net for Gulf pipefish. When a specimen is caught, Sunny quickly returns to the boat for data collection. When Sunny returns to the kayak, each pipefish is given an identification number. Then Sunny sets to work on retrieving a blood sample from which hormone levels can be determined.
It takes males in this species longer to be ready to mate again than it does females. So if a male and female mate at the same time, that male will take longer to be ready to mate again, and the female will be ready to mate sooner. So if you have a lot of females that are ready to mate and males that take longer, this sets up an inequality. And males become in demand, so females have to compete for access to them. And in that competition, you can see aggressive behavior.
With the first specimen captured and recorded, The day has just begun. Sunny hopes to collect 70 individuals over a period of 2 months. After a very successful field day with 8 Gulf Pipefish in the bag, Sunny and Jason head back to the lab, where much more work is left to be done. At the lab, the blood that was collected must be separated into its components using a centrifuge. Because the pipefish hormones are found in the plasma, Sunny collects only the straw to pink-colored liquid portion of the blood. Sunny also collects data on the animal's size and weight, which will be correlated with the hormonal data when they return to the lab in Oklahoma. By comparing the data sets, Sunny and her colleagues hope to find a connection between the pipefish endocrinology and its behavior.
But I think in studying it, we can learn a lot about how a female's reproductive physiology is connected to her behavior. And it may just be something that happens in pipefish. It may be something that happens in other sex role reverse species. Or it may be something that is in all female vertebrates, but a little more subtle in species that we call typical. And by studying a species that is in an extreme situation, we can get a good idea of what's going on and then compare it to other species where the mating system is a little bit different. And by doing this, we'll get a better understanding of the bigger picture.
Through an examination of other species, humankind has increasingly gained knowledge about people's physiology in connection with their behavior. In the lives of human beings, where sex roles are less defined, the Gulf Pipefish may be able to teach us something about our own physiology and how it relates to our behavior.